G'day. How you going? Good evening as well, if it's a bit dim where you are. This is a medical cannabis show, and I respect the law, and I hope you do too. Today's an open topic, so if you've got some cool discussions, throw them down, and I'll start throwing some slides, and we can generate some chat. Some cool discussions. Sorry about that. It's got YouTube opened. There it is. Yes, back to it. So nothing's really organised, but I thought about discussing microbes. So if there's no real questions come through, it's going to be a microbes discussion. Plant microbes, their interactions, what they do. It's how you can make them work for you, how you can make them, well, you don't make them, how by getting the right ones in your substrates, in your foliar, in your inside of the plants even, endophytes, you can make them do what they should be doing, or you hope what them be doing, which will create better outcomes for the plant. Cut a long story short. So there's different ones, like this. In botany, you can control with phytohormones, you can control different growth patterns, fruit size, all sorts of different things, shape. You can stunt things, you can use anyway. And with other microbes, you can do something similar. So I'll go into that a bit detail if nobody's got any questions, because it's pretty cool. Biofertilizers, that's a good word. So I'll hope that'll be in the title of today's show. If not, g'day GMF. There you go, mate. Sounds like fun. I like micro life, says GMF. Yes, microbes are cool. The more I get into them, the more I realise their full potential is not unlocked, because it's hard to get the right microbes for starters, and then it's hard to put them in the environment where they can thrive. So one of my courses this semester is Advanced Microbiotics. I think it's advanced. Yeah, it's Advanced Microbiotics, and it just goes into more of the patterns of the microbes, and that's what made me come to this topic of today's show, I think, because no one's really said much so far. And yeah, it goes right into detail about the different, the NIF and the NOD genes and stuff like that in the microbes, which is, say, for instance, nitrogen fixing. And if you can get that happening, you've got free nitrogen. And then there's phosphate solubilizing bacteria and all other ones that we can get that you can basically feed the plant for free, ongoing. And if you do organics, it's going to keep doing it. You'll have those endospores that will be dead from the microbes. Actually, I usually wait a smidgen. Look, see how excited I am about microbes. Wait and see if anyone's got the topic. Yeah, I could just start. I usually give it till 2 minutes to wait for people to rock up to see if there's anything. I'm just going off on it. It's because I'm going to keep talking. It's because they're so blooming beneficial. You can go to the shop, buy fertilizer, put it to the plant, bang, it's done, and probably really so minute amounts would be absorbed into the tissue. So a lot would be leached out, others volatiles off. But you can use microbes, and they'll do a similar thing and keep on going and doing it. Azotobacter, for instance. Azotobacter, or Azospirillum's a bit better. That is a microbe that releases two types of hormones, gibberellic acid and cytokinins. So that's nitrogen fixing as well, because it's azo. So you've also got fixing nitrogen, getting free nitrogen in the atmosphere and putting it into ammonia, and then nitrosomes can go and break that down into NH4, which is an uptake and it's a readily available source for the plant, because the two nitrogen forms that a plant wants is your NH3 and your NH4, your ammonium, and your nitrate, NO3 plus. Yes. So yeah, by getting the right ones. All right, looks like GMF's happy. Go, Aussie, go. Whoop, whoop. All right, I put out a video through the week, which was related to this as well, because I was just been going through it. Where is it? And it just talks about this, basically. So this is some examples of phytohormones producing bacteria. So I was saying before about azospirillum, gibberellic acid. So it puts out gibberellic acid. I thought it was its cytokines too. CK, there you go, azospirillum species. So it releases cytokines and gibberellic acid. So if you want to bulk up your what's going on outside? For instance, if you want to things are going to struggle, or you want to swell things up in late flower or in flower, day 25, you might introduce azospirillum lipoferium, and that's going to release some gibberellic acid, which is known to swell fruits. So your fruit at the end will be nice and swollen. What's another example? What else? Oh, that's right. How about Pseudomonas? Most Pseudomonas, see up top, Pseudomonas and Bacillus, they release indole abscisic acid, which is auxin. And auxin is really beneficial in various different plant growth development stages. And then if you also include some, where's Florensis? Pseudomonas florensis, no. I thought it was up here somewhere. Pseudomonas florensis is really good for fighting Pythium and Fusarium. So from introducing that species, you've also got the benefits of doing that, as well as your phytohormones. So it's some other ones in here. Well, everyone uses Subtilis. See, and that releases auxin. There's Florens up there. Go back to that. No, that'll do about that bit. I'll see if anyone rocked up. No one's really saying much. This looks like we're going to be into the plant microbes today. Why they're good. It was similar to last week, but I'll just probably elaborate a bit more. Share screen. So I just thought I'd throw it into the search engine and see what come up. So before I go through, this is what I just put into the search engine to see if there was going to come up anything rad, but I haven't actually been through this yet. Doesn't look like anything good. And here's Wikipedia. It says, plant microbiome, also known as the phytomicrobiome, plays the role in plant health and productivity and has received significant attention in recent years. It plays a lot more. It helps with resistance. It helps with the hormones. It helps with everything to do with its growth and development, different production stages. It's like by putting in that, what's that one that produces gibberellic acid? Some azospirillum. By putting in that one, you can get swollen fruit at the end, as an example. So it's a lot more than people are. It's really rad. So I'm putting a lot of focus into this. So I haven't looked at this even yet. Yes, doesn't do that. Oh, the different stages. Is this beneficial? In your substrates, there should be a 50% difference of your beneficials to your pathogens. And because of your good plant health and its immune system that it has, it can decipher the good and the bad and stay in good health. Environmental factors, phytosphere. Phytosphere is above the ground, see that in the blue, and rhizosphere is actually right next to the roots. Cyanobacteria. It's another example of it says, it's a nitrogen fixing bacteria if you get the right one, Anabaena or Nostrec. The one I use, I'm not going to go into that. But yeah, so that's another example of some nitrogen fixing. But they're they need light to activate. That's cyanobacteria, blue-green algae. So that's not really helpful. You can do that, inoculate plants, pots, sorry, that have clear pots, you know, they're normally black. If you get them and put clear, I've gone and got some clear ones. I've recycled some milk bottles. So you cut the top off and keep it as a good little shape, and you still reuse those. You put some of this next in it, and it's nitrogen-fixing cyanobacteria, and it sits there. It's green on the outside of on the inside of the pot. And what it does, that green is the chlorophyll, and that can break down to be readily available nitrogen for the plant. So that's also free. Yeah, that's something I actually do. Let's go. I want to touch on the endosphere. No, it's inside. All right, the different regions of it. What's my soil science? Let's go into that. So this is on some biofertilizers. I haven't declaring fertilizer response. I'm going to skim through a few. Oh, so release. No, we're going to get into the biofertilizers. Fertilizer use efficiency. Yeah, it's so low. I'm not going to stay on topic. This is a really good course because it taught me about how to use fertilizer and why and when, and all the, like the slow release, for instance. It's to do with the size, the granulation size. Like if you want to make it really extra slow release, just bulk it up. But biofertilizers is what we're talking about today. See, that's the NPK of using different organics. So you can get really good outcomes by just doing that. So yeah, since learning all this stuff, it's taught me that organics is so much better. It's just very labor intensive, and a lot of people don't want to do that. But I'm a bit anxious anyway, so I don't mind putting in the effort. Effects of customized fertilizer on leaf crop. Yeah, then the research also shows that it's good. Here we go, some biofertilizers. Oh, this is a smart fertilizer. That's just actually, we're not going to go into that bit. We'll talk about microbes. Zinc oxide, no. Plant growth biostimulants. Biostimulants are in the microbes too. They've also put out different biostimulants for the plants. Growth hormones, enzymes, all these things can come out from the beneficial microbes. Fortified fertilizers, no, I'm not talking about that. Water-soluble fertilizer, that's very important for your irrigation. Foliar, no, not that today. That's a great one. They haven't got that in Australia. There's a nano urea that they've made that they've released in India, and it's not available here yet, and that's blooming excellent. I tried to get some water-soluble fertilizer. No, come on, where's the biofertilizers? I hope it's blooming in here. He's sitting back going, look at this crazy bloke. He doesn't know what he's on about. That's right, I don't at the moment. I don't know where I'm at. Fertilizer. Come on. Yeah, adulterated. That's what you've got to be careful about too. Where was the adulterated? Where people put in, probably not more so in our countries, but in other countries, if you're trying to source this material, be careful of what they put in it. Like with I'm trying to get some Azos Brillium, for instance. It's pretty hard to get, and I can get it from certain countries, but I'm worried about what their carrier product of it is. Like it might say 10 to the power of 6 that it's colony forming units on the front of it, but it might be 10 to the power of 10. It might be really low. So, and they might put a lot of adulterants in it, which make it bulk it up a bit. That's the word I was looking for. So yeah, you've got to really be careful about that. Adulterations of it. There's all different types that they do. See, they put sand, salts, all sorts of different bad things. I'm not going to go into it. It's major problems. Major problems. Weakness, no. Where's the biofertilizer? Oh, it's the end of the slides there. Rad. So it wasn't biofertilizer section. Ah, here it is here. Sorry about that. Here we go. Crop nutrition through biofertilizer technology. Biofertilizers are living microbes that enhance plant nutrition by mobilizing or increasing nutrient availability in soils or your substrates. It doesn't have to only be in soils. All these microbes can be used in even hydroponics, but they might just act differently because they'll be looking for oxygen as their electron acceptor, and it mightn't be as available, so it mightn't thrive as much. So this goes on to say various microbiotaxa, different types of microbes, including beneficial ones and fungi, are currently used as biofertilizers as they successfully colonize all parts of the plant, inside, outside, and right around it. And the better you can get, those spaces are going to be taken up anyway. So if you can get all your beneficials taking those spaces up, wow, isn't that rad? They'll all symbiotically just work together, and this will do that, like Azotobacter does nitrate, NO2 to NH3, and then Nitrosomus will convert it from NH3 to NH4, things like that. So if you can get them all working well, here we go, nitrogen fixing from the air, your azos. So they just sit there just for free, just as long as they got the right moisture, they'll be working because they're looking for nitrogen from the air to complete their growth and development. Benefits of biological nitrogen fixing bacteria. This shows in legumes. Oh, and that's by Rhizobia, which is a different one, which doesn't work with medical chem. Here we go, Azotobacter. Rad. To group up their oval. Okay, that just describes them. Several species. Cucurum. Okay. Dominant in soils that inheritable of fixing nitrogen. Yeah, 2 to 15 milligrams per gram of soil. That's heaps. Mechanisms on non-symbiotic. This is the azos. So this is the one that's really important to us. So yeah. So see, it goes from N2 into NH3, and then you got azospirillum taking it from NH3 into NH4, which is readily available for the plant. Excellent. Well, thank you for that. It worked out quite well. Formulation of large-scale inoculants. Oh, okay. Of course, you can inoculate your seeds and other things with it, and you'll get rad outcomes like azospirillum. That was the one I was talking about before that has the gibberellic acid and the cytokinins. It's non-symbiotic, which is what we want. Associated nitrogen fixer, aerobia. Okay, good. Introduce production, plant hormones, antioxidants, osmotic adjustments, defense strategies such as the expression of pathogenesis-related genes or PR genes. So it primes the genes, getting them ready to activate the immune system when it comes under attack. So that's pretty important in your genetic line, because otherwise it'll sit there, and by the time the immune system's activated, that your problematic pathogen has taken over, and then it's too late because the plants can't do much because it's too huge. Thanks. Azospirillum brassiaceae promotes increase in growth and yield. Shows this experiment, and it does because it puts out the cytokinins and the gibberellic acid, 2 phytohormones. Right. Cyanobacteria, that's the Anabaana and the Nostoc, which is the 2 really cool ones. but they require the light to survive. So it's hard to, that's why on the outside of the pots I've found, and clear pots, it's really advantageous. People don't really talk about that. It's always got to use black pots. Well, why not do this? Have free nitrogen and use clear pots. Oh, wow. And it works. Cyanobacteria. Sustainable agriculture and environmental development. Yes, it helps. Win-win everywhere. It's so cool. So I'm going to go through with its bonuses and things that it does, because I've already talked a bit about them. There's a lot more to talk about. Phosphate mobilizing and solubilizing microbes. Mycorrhizae is an example of that. Actually, I think Bacillus subtilis is a phosphate solubilizing bacteria as well. Association. Types, no, I'm not going to go vascular. No. Okay. Simplified flow chart of biofertilizer. So you inoculate. Isolation of microbes from the soil. This is if you want to make it, but if you want is to best is to buy it. So you can see, and it has to have the times 10 I was talking about last week, the colony forming units. You want the highest amount. So in the billions, you know, would be really nice, or in the millions, at least it's going to have something. And if it doesn't say anything, like, you know, I can suppose I can say root roids, that doesn't say anything. So in other words, you've got to use heaps of it, because it's so diluted that by using a little tiny bit, it probably won't work. But the benefit with these biofertilizers is you put them in teas. So you just put a little tiny bit, maybe depends on, see, again, it depends on the colony forming units, but you can put a way smaller amount in a tea and let it sit there for 36 hours under oxygen with the right temperature and bubble away, and depends on what microbe it is, and it can increase its numbers by giving it what it wants. Like just by putting molasses in, it might be a good carbon source, and that's taking care of that one side of it, but it mightn't be the whole other growth and development sectors that the microbe needs for it to thrive. It's a good way to start. It's a good thing. That's how I do mine anyway. Maintenance and mass production. No, I'm just talking about that actually. That inoculating it into substrates. That's what this is about. So they just inoculate it, put some in, sprinkle it over, mix it around, and let it do its thing. And then before you know it, you've got a large amount of that microbe has colonized into whatever you've inoculated, if you've given it a good amount of moisture and temperature, and oxygen more so. With all the microbes that we want to do with oxygen, not the anaerobes. So it's this isolation and selection of phosphate solubilizing microbes from your soil. The composition of that media is as follows. Okay, yeast extract. So that's like they're going to grow it. Oh, okay, that's right. They're going to separate it from the soil. Yeah, so if you, by not giving, say if you want to get your nitrogen fixing bacteria out, by not giving it nitrogen, and it's going to pull it out of the air, you're going to see which section grows, because that's going to be the one that's going to do that. So that's an example for the nitrogen fixing. This is a bit more detail of the phosphate solubilizing ones to pull out of the soil, because you've got to eliminate or put in the phosphate and see what the different reactions are for it, see which one grows more and that sort of stuff. But a better source is to go and just buy them. And then you're going to have houses for them to live in, like your biochar or organic matter, your chopped up sticks. So once you get all your microbes, you're going to have somewhere for them to keep on going. And then you can look after your microbes. So buy your compost and mulch, and really, really concentrate and look after them. And that's what I focus on. Structure of, right, this is off the bio, so the next one is mulching. Oh, this will do. So by looking after the microbes, they'll look after you. And this is one of the ways that you want to do it, with mulch. And make your own, of course. just by letting it break down. Put big heaps of organic matter, all your seeds, everything like that, because remember it breaks down if it gets, when it should be in the thermophilic range above 65 Celsius. You can see it, you can test your actual compost heaps by looking at them in the morning when it's really sort of cold outside, and the heap should be warmer, and then you should see a little heat waves coming out from the top of them. And that means that it's activated enough. And if you don't see that, try putting in a few 5 gallons or 20 litres of water or even tea through the whole pile. Well, it depends how big your pile is. And I like making piles that say 2 square meters, 2 meters divided by 2 meters, or what's that? 6 feet by 6 feet, because otherwise too big, other bigger animals can get in there. Anyway, I'll get off the topic a bit. So yeah, look after your microbes. Mulch is one way. Should see if there's any. See if there's any questions. Oh yeah, a few people are in here. Micro fun, yes. G'day, Aaron. Nice to see you, mate. GMF says always get a kick out of algae growing on my pumice and perlite. Cool shades. Make sure it's the right ones though, because you can get yeah, that's because I saw it too back in the day. I thought I want to get the right one, so I went and sourced Arthrospira platensis and cyanobacteria, and that's the one I used. That's actually, that one has tryptophan in it. It's 75% tryptophan, which is the biocursor, the pre-element before oxygen. So by having that heavily in your substrate, the plant only has to do one little thing, or the microbes have to do one little thing to switch it and change its form. So it's pretty cool. Aaron says, my now 5-year no dig beds are finally starting to work properly. See, there you go. I have the biggest and best tomato bushes I've ever seen, and best part that haven't watered these garden out to sea. Brilliant, mate. That's exactly what big pharma and the marketing people and all that sort of stuff, they don't want you to bloom and do that, because you're not going to pay for all of their nutrients or their pesticides. So by you just doing it that way, you can work on your microbes and let them work for you. And then you can also get your bio pesticides and inoculate that into the system. And before you know it, you'll have some little beneficial mites running around that are rad, where at the right time, they'll just shine their head and bang, they'll take care of any problems that arise. So it's really, really simple to go forward in the organic sector. But it's labor intensive, and but once you're there, like Aaron's blooming there, and he'd be sitting back just rubbing, like GMF says, yep, well done, Aaron. Exactly right. You're sitting back, mate, just rubbing your hands together, going, gosh, this blooming is easy. People around you going, far out, it's so hard, I've got to spray this, spray that. Yeah, because you're doing it the wrong way. Microbes. Aaron says, I'm now getting many tomatoes that are also the best tasting he's ever eaten. That's right, because you've activated the secondary metabolite profile from all your microbes. They've allowed it to happen from that growth balance differential hypothesis. It's brilliant. GMF says labor can be circumvented with good planning. I will agree that organics are actually less work once you understand the process and all the work's done. Yeah, after a few years of getting into the cycle of things, it happens. It lessens off. Well, that's how the old farmers used to do it. They just used to go and water and maybe top dress this and cut that down and farm. Well, it's only these new modern day farmers that want to employ less people but pollute the environment. And now they're going, oh, we can't change this sort of stuff. That's right, because it's too costly for them to go back, because they're getting their good price per tonne or whatever for the farmers, for instance. And if they go and change stuff, they're going to drop a price bracket. And all of a sudden, that's going to take them out of market. So that's why a lot of the farmers who grow synthetic are going out of the market, because they can't afford to go back and repair everything. I didn't complete that. We just, GMF complete by saying, we just feel more in control in a synthetic mindset. Yes, that's right. It's quick. Everyone wants it fixed right now. They want their plants to be green. Quick, put nitrogen on it. Oh, look, 2 days later. Oh, wow, look how green they are. That must mean they're fertile and extremely healthy. No, that means they've absorbed the molecule of nitrogen. That's all. And if you force-fed it to them, they've just gone and accepted the NO3 or NH4 without it coming from a microbe, or without the plant having to ask for it and use. So that's defenses is actually lessened. So, yeah. I went to I'm going to solve that. Aaron says most of his garden has got a layer of mycorrhizae, and now I have many different mushrooms all around the garden. Brilliant. And then Aaron says, let your garden grow. And then grow, it might kill the grass underneath if it's really, really long. And if it goes to seed, anyway, I like that idea, mate. It is. You want to use don't use a catcher. My mum's in a bad blimmin' way and still uses a blimmin' catcher. I tell her so many times, but I don't want grass under my feet. Righto. Aaron says it has taken 5 years. Only pull the weeds you don't want. Lots of weeds are actually beneficial. Free cover crops you can just chop and drop as green mulch. Well said, Aaron. He's into his organics, doesn't he? Yep. The green cover crop. I'll just stick on topic. I was going to go into agriculture then, but talking about green cover crops. Yes, well, that can be. What was I going to say about those? Putting that in, you want to cover it up pretty quickly, because it gets changed. A lot of these microbes can convert it to NH3, which is volatile. It's ammonia, so it goes straight back into a gas form, which goes out. Wind comes and bang, it's gone. That form of nitrogen. So if you cover it, you've got a chance for nitrosomes to come in and grab that and work with it and change the form, so it'll form a cation, NH4, and then that will stick to a molecule which is negatively charged, and it'll sit there and be bound up. Or a microbe will take that in, a part of its nitrogen assimilation process, and it will be immobilized inside of it. So when that microbe breaks down, that form of nitrogen will be readily available to the plants. Yes. Jeez. All this it's off the top of my head, all that stuff too. It's really good. All these studies, it just pays off. It's terrific. Aaron says, organic only is the best nature can offer. That's how we've got to this date. And by people not doing organics, we have to use bioremediation processes because of all the mines and the toxic pollution that's been created. It's sad. Look at China. Most of its substrate, most of its land is inoccupable for growing. Even under the States, USA, 95% of their groundwater is polluted with pesticides. It's really sad. And from learning this stuff, I would like to help more people about it, but most people aren't interested because it's too labor intensive and costly. They're not willing to go through the steps to do it properly. Yeah. But I am, and so is Aaron and GMF and all these other cool listeners, because you know which way you can go. And it's about being sustainable. So building that down the track, you want it to be ongoing. And like Aaron said, he's already gone and developed his gardens so that they've been organic, and now they're taking care of itself. So if you go and get like, I've gone and got some Bacillus thuringiensis, and that's a rad microbe that works in defenses. There's that Bacillus florensis, that works against Pythium and Fusarium, and also produces a couple of hormones. There's the, I ordered 4 the other day. I have to look. Azotobacter, I ordered that one too. Free nitrogen from the air. So that's just going to sit there and multiply in the substrates forever until you poison it or eliminate it like that, because it's endospore, and that's just going to carry on. What was the 4th one? Bacillus it's hard to say it, ligiforium or something like that. And why did I get that one? It's a biofertilizer, and it works with, I can't think off the top of my head. Let's see if I can. Anyway, it's beneficial too. So it's getting them to work for me. GMF says, when he breaks a fresh branch and use it to scrub the inside of your pots, to the exposed part of the branch will turn purple. Okay. I read that this is a result of cyanobacteria reacting with starches. Thoughts? When you break a fresh branch of what? Of mycelium, yeah, I've heard that. And that's when its active ingredient, psilocybin, reacts with the air and it turns purple. I've never heard of that with cyanobacteria. So you're thinking that cyanobacteria is an endophyte inside of the branch, and when it snaps to get oxygen, it turns purple. Oh, when it reacts with starches, it turns purple. Okay. I can't confirm if my cyanobacteria is inside of the plant. Platensis, I know it's on the outside. I haven't done any culturing techniques where I've snapped a branch off and then cloned it and then seen if it could produce in a clear pot again to prove it. I don't know, mate. That's a really good question. Good on you. So when you break it, oh yeah, it says down the, what's this? You don't wash your pots. You always leave residue to try and keep my bacteria and other microbes. Be careful if pathogens are multiplying in there. If it's like an anaerobic spot in the corner of it, like in the bottom of the pot, where it might have been not breathing, like in those little crack things in the bottom, where it just might have stuck there, for instance, and that might have been an unoxygenated spot, and that's where the anaerobics might proliferate. So, yeah, maybe look out for that. But otherwise, yeah, why not do it that way? Don't wash your pots. If you've got no problems with the grow, no disease, infections, anything like that, why not just keep it going? And then you will, like GMF says here, your bacteria and the spores will be, because they'll mostly be dead, so the spores will be there, just waiting to act again. Same for using your roots. Good idea, GMF. Aaron says, is there somewhere that can test for all that in your soil? Elaborate. Because, yeah, you can do it. You can do it yourself. You do it by pulling one microbe out, like by pulling out, if you want to pull out your nitrogen-fixing bacteria from your soil, you can isolate it with a culture method and don't feed it nitrogen. So it'll be looking for nitrogen out of the air, and then you'll see that one proliferating, as an example. So, yeah, is there somewhere you can test for that? Test for what in your soil? Yeah, that's what Aaron's going to get back to us on. GMF says, can a branch? I use stems to empty pots and refill them. I think it is in the soil, but when I use broken stick to manipulate soil, it starts turning purple. Wow. Interesting. I don't know, mate. You can put your stem in a culture, like in an aerocloner, or just clone it, and then see if you've got it in its endophyte. So then if you see it's back, then you'll prove if you have got your cyanobacteria as an endophyte in your plant. So that would be a test method, by cutting the branch off with the purple and then cloning that and then putting that back into a pot and then seeing if that cyanobacteria did proliferate on the outside of the pot, possibly. Something to think about. That's pretty cool. GMF, I like you. He's always into his science stuff. It's really good. Just noticing what goes on. You don't have to be hardcore and have a full-on lab with all massive $100,000 machines. You can just notice and look what's happening. And by these little morphogenic expressions and by different branch snapping techniques, you can really get some good science starting to go on. So good work, GMF. Oh, here we go. He goes on to say he has an open bottom container. They get a lot of air and the roots in the root zone. Oh, yeah, nice. Yes, it's very important for that, because that's where all of the oxygen gets, the plant gets most of its oxygen out of its 17 beneficial minerals, elements. 95% come from the air. Oxygen's 45, carbon's 45, and protons are 5, actually there's 6%. And the nitrogen's 1.5 under that. So if you can give it the conditions that it requires, you've half won the battle. And by GMF doing that, he's blooming winning the battle. So you'd notice, when you give it a lot of oxygen like that, I've found that the stems get bigger, or no, actually I can't really remember, that was a while ago. But I found some good, using those aeropods, I found different things were happening to the plant because it was getting better oxygen. It's like Liebberg's law of the minimum says whatever the least thing is, if you've got a low amount of nitrogen, that's only as good as the plant can be. So its most limiting factor is about the best that the plant can perform. Aaron says, microbes, bacteria, and nutrient soil test. Is there somewhere that does soil testing? Yes, you can do it. Oh, if you've got a compound microscope. You need a hemocytometer, and that will allow you to count the microbes in your soil, in your substrates. And then at that, you go, well, what the hell do I have? So you need a metagenomic test to see which, so they put it through a 16S RNA sample test, and that tells them, because all the microbes have 16S RNA and humans have 18. So if you put it through the 16, it'll come back and tell you which microbes are on the list. Cut a long story short, once you run it all and then put it through its database, and it'll come back, and that's what the result will be through a metagenomic test. So that's how you find out exactly what you've got in your substrates. But in general, you can do it by looking under the microscope, and a lot of them go off shapes. And let's actually, Aaron's asked the question, so let's pull that up. Open a new window, microbes. What do you call it? Identification. So it's the shapes. So where is those slides? I'm just looking for, I suppose I should share it with you guys and girls while I'm looking at it. I'm going to have to go in. I'll start at the back bit. Oh, I'm not sharing yet. Sorry, all this clicking, I have to get back and share. Kind of forget what I was talking about now. These are examples of the different microbes in different regions of the plant, inside and outside, bacteria and fungi. Oh, that's right. We're getting back to the microbes and identification. Because, yeah, see, this is to see what sort of colony-forming units you get. This is 17 billion colony-forming units. it's quite a lot. There you go. So all these people, to make these up, all they do is they source all these. Like I'm getting Ligoformis, and I got that one, Symbiotic, and then they just mix them together. Megatarium is really expensive. And Amy Locophacians, it's good for abscisic acid, which is actually, it's a drought. It's not a good thing for the plants. So I weren't real keen about that one. So by working in with the ones that you want, oh yeah, they're the fungi up the top, bacteria in the bottom. But anyway, let's get back to it. We're talking about the identifications of them. I'm looking at a slides that comes up for that. Phytohormones by plant growth regulators. That's what these are. So you can have beneficial, like PGRs. You can have bad plant growth regulators, and they're ones that are harmful, carcinogenic, like in the brand. I'm not going to say it because it's bad, and they use it in their formulas. There's a couple of them in the hydroponic formulas. Oh, look, the NIF genes. That's cool. The microbes have got about 8,000 odd genes in them. Then it goes up to the plant. Our plants have about 30,000 genes in it. Pairs of genes. So if you can manipulate or learn what they do and try and, like this is the nitrogen fixing gene. So if you can engage this one and activate, how do I activate this one? Or how do I make this a dominant gene? You know, as that sort of stuff. You'll be really on the winning. You'll be a win-win. I'm going the wrong way too here. Yeah, phytohormone production. If you can pull all of them out of microbes, why not? That's what I'm into doing at the moment. I saw fluorines there, yes. Pseudobaton produced by Pseudomonas fluorens, and it works. Oh, that's right. It's an iron. Cytophore is a excrement put out that's iron chelator. It has high amounts of iron in it, and fluorens does that. Okay, cool. So that's the one I've ordered. This one. That's also the main reason I ordered this one is because it works extremely well with Pythium and Fusarium, and those two fungal diseases in substrates are so hard to get rid of. If you're in an environment that's just going to overwater or humidity is too high, substrates aren't designed. So if substrates are designed poorly, like too clayey or something without enough. I should go through these later instead of reading these with you guys. I'm looking for, there was a good soil one before about the bottom washing of your pots. If you see a soil particle that's half an inch wide, there's a chance on the inside, if it's clayey, like this one, it might have anaerobic spots. I think that's pretty cool. Microbes identification, come on. There's the classification of them. Those big long words, 5 major nutritional types of microbes, bacteria in the environment. Yes, I'm not going to explain. Come on. Identification, please. No. That's how to test them. If you want to check your serial dilution rate, we're talking before, if they have 10 to the power of 6 colony forming units, you go, all right, let's see, do I divide it 6 times? And that means I can count them. So you'll do serial dilution, so 100 mils and 10. So into 90, 10 into 90, 10 into 90. Or you can just drop it off 10 mils and put 9 mils and drop 1 mil across. That's a bit high. But they're doing soil up the top. So that's if you want to count everything in your soil, you'll count your dilution rate and see your amount of microbes. So after you do your serial dilution, you'll put it to your hemocytometer, if you've got one, and then you can count the little squares underneath it. Or if you haven't, you just put it under keep diluting it until into the plates. And then you can count the colony forming units in that, because you'll do a swab here and then you can count dots. So if it hasn't got dots, it means you'll have to dilute it again. Come on, identification. I'm looking for the squares. Here's another way. You asked before how to get your own microbes if you want to isolate nitrogen-fixing bacteria from the soil. So you just do it this way. I'm not going to go right into detail, but you want to, out of the 4 plates, 1 doesn't have nitrogen in it. And that's the one that you'll see that will have nitrogen-fixing bacteria, because these ones have that added into it, so they don't have to make nitrogen. But this one hasn't got any, so you'll notice that this will probably have a lot of nitrogen-fixing bacteria actually in it. Well, there we go, bit of identification, isolation of anaerobic bacteria. No. This is the way to do it with the 9, put 10 mils in the start, take 1 mil out and add it to 9 mils, and you can keep going to see what growth it is. And you'll see here, as this example, they've got no growth in the -5. So you know that it's 10 to the power of 4 is this range, this colony forming units that you purchased. And then you'd also do a Petri dish out of this, because there's no growth in here. You do a swab out of this and it might come up with 10 or 20. So you know, all right, 10 or 20 to 10 to the power of 4. Isolation culture, no. That's how to make it. That's all the different, you want but before on how to make it. This is all different ones to using the elements that it needs. So you can make that bacteria proliferate and therefore get more of that bacteria. Oh, nearly shapes. Yeah, the availability of nutrients, macro and micro. So a microbe still needs all of its nutrients to create its growth and development. Amazingly, but they're very in great details. Like ferric microbes require iron. Nitrogen-fixing bacteria require nitrogen. So they all require different elements in major amounts. Yeah, these is all the different things that a microbe could do. So I can fix all these different things in the atmosphere, in the substrates. Like that's how Mother Nature survived. Gone through all these volcanoes, all sorts of real harmful, toxic events, and they've all been fixed. And it's by microbes in different ways. These are the different ways that you can make your microbe work for you. Life under highly dynamic conditions. It's pretty rad, eh? All the different forms of nitrogen on how it happens, how it cycles from this to that. It changes from NO2 to N2, goes back to NH3, and all these in the middle are pretty much microbes doing all this process. So you get the right microbes, and you can transform this into this. And so you're looking at a lot of NO3 producing and a lot of NH4 producing, ammonium producing, which I don't see there. Oh, because this is it's not the ammonium. Auto control. Here we go. Here we go, Aaron. So under your microscope, you want to see these types of things. So they identify it from shape. And Bacillus and Pseudomonas are pretty much look like a tic-tac. The coccus is round. And the coccus, this is Bacillus, so it's a bit longer because coccus is round. The spirochetes looks like a corkscrew. And Spirillum, like Azospirillum, looks like a flattened out corkscrew. And then fusiform, these things, I didn't know what these, I had a lot of these back in the day when I used to, because I got a rad compound microscope, I used to put it on, do it all the time, which I should blooming do it. I'm talking about microbes so much, I should do some examples, and now I know a bit more about it. And this is some vibro what's that one in? Vibro disulfur. It's a few notches down the scale. I'm not going into detail like that. But there's a way to identify them. So it's pretty much just by their morphology shapes you identify the bacteria under a microscope. Because to know what type of, is this Bacillus subtilis or is this Bacillus thuringiensis? Who blimmin' knows? You'd have to do the DNA test to see what it actually does. Or you could put it under the test where, say if it's those two, Bacillus subtilis or thuringiensis, you could go and put it into the conditions that one of them really like and see if those numbers proliferate. But again, how are you going to identify it? Oh, you'd identify it because it's proliferating under the conditions you've created. So it's kind of a bit of guesswork again. So, yeah. Thank goodness for technology. They don't just identify them from this anymore. That's why the world can really, they're getting into microbes now. It's more a specialized thing because the technology has allowed for it to happen. Yeah, I like this slide. Look at that one. Isn't that cool? All their eyes and that. Look, there's the coccus. You can tell me some of the shapes now. Go on, Aaron. Yes, this big. Yes, this grainy looking one. And there's one that we all know, very common. Yeah, it's pretty cool. Protozoa. That's actually one of the sections under the advanced microbe course, microorganism course I'm doing or starting soon is protozoa. So I'll be sharing all those results with you. This is Azospirillum. So this would be plating it. So see how they've done a strike here. So you start by wagging that way, then you finish off by going upwards. And there's a bit too many. So by the time that's the little round needle thing that you use to dip and put the swabs on, it's gotten right to the end and it's just run out. So you can nearly count these, but it's not good enough. So then you go for another serial dilution into here and bang, you can count them all. Oh yeah, and that's the microbe, and that's the endospore, one half of it. So see that bit there? That might be actually dividing through horizontal gene transfer. That's how one of the ways that they can proliferate. Yeah, I won't go into that. That's right, the shapes. I was looking for any type of more identifications. How do you identify microbes? Being at the microscopic level, it's really blooming hard. Well, pH is one good thing too for identification, because if you've got a substrate below 5, you know your bacteria might be in spore form, but it's not going to be doing much because it's not in the favorable conditions. But on the other hand, fungi really like it below 5 pH. Maybe another identifying thing. Yeah, temperatures are another identifying thing. Evidence suggests that these 4 species bacteria act together as a biostimulant. Yes, this is one I look to get. Enterobacter cloacae, petita. Common amonis. Never even heard of. Testosterone. Okay. Oh, that might be really good. Testosterones. But yeah, see how this is a good slide because it says working together. That's the key thing, where quite a few microbes don't. They are, what's an example? What's that lactobacillus? What's the one that everybody uses? Korean natural farming where they use lactobacillus. It's a really heavy spore creating bacteria. Biofilm creating bacteria, and in that biofilm, that put out a lot of antimicrobial anti-resistance, so that if other microbes come into it, they can't enter past that because it's got too much properties that they don't like. So that's an example of not being symbiotic. So by chucking all your microbes in and going, wow, I've got the rad substrates, that mightn't be the case because there might be some working against the other ones. So you've got to get the ones that work with each other. Like lactobacillus is the heavy, ever since learning that, I've stayed away from those What is it? It'll come up. Unless, I should see what people are saying. See how I get excited with microbes? Oh, Jesus. GMF. Oh, that's GMF. That's how Aaron says. Oh, yep. Oh, yeah. I'm just reading the chat. And the quality. Yes. But the... Wow, yeah. Embrace nature. Fighting is losing battle. Yes, we do. That's right. All right. We're at the hour. What other microbe stuff could I elaborate on? I think that was about the only identification stuff that I got from microbes. I can maybe go through some more microbe courses I've done, but I want to keep it today on biofertilizer. Yeah, strictly on biofertilizers today. Here it is. Here's Bacillus ligaformis, plant. I'll just show you actually instead of telling you. Because what's he talking about? How do we know it's truth? Well, I'll just read it. Remember I was wondering before about Bacillus ligaformis? Well, it's used for a growth protection and biocontrol. So after reading this paper, this is what made me include it into my microbe circle. And yeah, so I just went through and I can't remember in specific. Okay, testing the genes. Oh, that's right. See, biofilm formation. Anyway, that was it. It determines, they test the amount of oxygen that it produces, which is a plant phytohormone, and it helps with identification, phylogenetic activity. Yeah, it helps with anti-pathogens. Nitrogen fixing. Does it have any nitrogen fixing properties? Yeah, well, does it? Yeah, it just says to examine. The experiment was done thrice and triplets were made. Does it have any phosphate solubilizing activities? Where if there's phosphate sitting in the substrate, this bacteria comes in contact with it, will it actually break it down into a form that the plant requires? So yeah, it was a pretty good. And there's the site of the iron. Does it do iron? Is it an iron chelator, or does it break down potassium? So it's handy from reading these things. Was that what I was looking for? Phytohormones, bio-nitrogen fixing bacteria. Let's see what's under this one. Here we go. No, where is it? Oh no, let me go through that. Does it say any types of microbes in the cycle? No, it doesn't. These are some different ones. So rhizobium's not, it's with legumes, so it doesn't work with our plant, unfortunately. But Brady has a section where it does, and then it goes on to some other ones. But if we stick to the rhizos, you can't go wrong. And this is actual, breaks it down into a species form. So then it's got its genus there and its species right next to it. So then you can check on this paper and you can see on what individually it does. So if you've heard there's a real rad one, that I'd try and go with the ones where they produce phytohormones, because having phytohormones, you've got it's hormones in the plant, and it can decide if it wants to use it or not. And you know from having hormones in athletes what happens by giving them enhancing things, all good stuff happens. And yeah, symbiotic. You want non-symbiotic, because the symbiotic ones pretty much cover all of the legume type of ones. Actinomycetes, cyanobacteria. Weeds. I see some questions going down. I've been making my own biofertilizer for a couple of years, Aaron says. You can use it in full stink time of the year after only a couple of months. But leaving it sit for a year or more, all of the stink goes away. And now, Aaron says, he has lots of beautiful black non-smelling liquid compost that the plants seem to like way better than before the stinky brew. Well done, Aaron. You've used your nose to decipher the geosmin terpene, which is healthy. So by going in it, you'd be smelling all those crappy ones. You'd be smelling off gases, sulfur, and all other antibiotic, anaerobic, biotic microbes that don't live in the air, unoxygenated microbes. You'd be smelling those ones off gassing from their breaks down of those materials. And now, once they've broken it down, it's all gone, everything's stabilized, and you've got the pure geosmin smell, which is what you want before you give it to the plants. So you know that everything's decomposed enough and should be in the form ready for the plants. Well done, Aaron. He knows. So non-symbiotic is the ones we want. So azos, Clostridium, no, don't use that one. Cyanobacteria, yes. Clostridium is, don't source that. That's for blooming, that's what you get in anaerobic conditions where there's no oxygen. Yeah. Remember, this courses are done, these aren't just for our plant, this is done for all plants. So it's hard for me to go through and pick up the ones that are really, that work well. Azotobacter, Spirullium, Azospirullium. Those two are the ones that are good. Plant growth promoting rhizobacteria. It's a section they come under. Nitrogen fixation. Oh yeah, here's how it happens. I was saying before. So if you get the ammonium in the sense of oxygen and with nitrosomes, it'll change it to NO2. And then if you get NO2 with some oxygen, Nitrobacter will change it to NO3. So by having both of these microbes, you're going to get free plant available nitrogen. Denitrification. That's another side of it. Anaerobic. No. Enrichment culture. This is kind of what you're talking about before, about making your own nitrogen. So here's just another enrichment culture where you can go and do it. So I'm not going to go through it because I kind of explained it before, but that's another one that you can do to get your azobacters. See, again, you don't know if you're actually isolating those individual azotobacter. So it could be azospirullium, because it could be a nitrogen-fixing bacteria. But then you're going to go and look at the coccus, put it under the microscope. And the cells of azotobacter, Coroclium, are the coccus cells. So that's them there. Azospirullium is a free-living non-symbiotic, does not form nodules. Yes, that's right. And it looks like that one. Oh, okay. See how it more looks like a bacillus or a pseudomonas. So it's just, yeah, it's really hard to identify under the microscope. But if you've pulled this out of and done this enrichment culture with looking for these, you'd know by looking under here, you'd be pretty much on the money. And look, they can fix 10 to 40 kilos per hectare. That's the amount of, like 40 kilos per hectare might be the or you might be looking for 100 kilos per hectare of nitrogen that a plant needs to complete its full growth system. So by getting half of it from one microbe, wow. Oh yeah, Azospirillum produces auxin, disease resistance, and drought tolerance for some additional benefits of inoculating and using Azospirillum. Brilliant. It's free ongoing. For those organic folk, this is such a win to have these in your blooming substrates. Never kill them off. They're always going to be there. Not going into that detail. There it is. Is that, what scale is that in? I think that's 200 micrograms. That might be, you could do that under the microscope. I think mine goes down to, yeah, mine could do that. Mine's 100 nanometers it goes down to. Not, and that's electron scanning. I wouldn't get that sort of definition. Clostridium. It looks like the blue-green cyanobacteria. Azotobacter. Wow. I'm going to have to start having a look at these microbes I get under the microscope. Yes, that's free. If you ever heard people say you can get free nitrogen from lightning, this is the formula. I'm not sure how it produces it, and the answer is it does. But we're talking about bio. There's a nice slide. Ammonia from nitrosomes into nitrate, and then nitrobacter into nitrite, sorry, and then into nitrate. So we're having two microbes, the plant gets fed. And ammonia can come from releasing, what was that one before that releases NH3? So if you get that microbe before it, it releases NH3, then you've got all these. So in other words, that would be symbiosis. So if these could work with the next one, that's symbiotic. So by getting this and this together, that's going to complete this process for free nitrogen in the form plant available nitrogen. Nitrococcus. Okay, nitrate bacteria. Oh, nitrite bacteria. Nitrate. So there's only nitrobacter for this one, but they found three that can do nitrate. Produce nitrite, nitrosomus, nitrospira, and nitrococcus. So in fish tanks, this would be, that's probably why they found that out, because they've done a lot of fish investigations with their nitrite, because that's why what's that system where they have fish are feeding hydroponics? This is the reason why, because fish excrement is nitrite. So if you have that, a lot of the bacteria you buy from aquarium shops will be exactly these ones. So you can go and source them there. So then all you've got to source is the nitrosomus, which is pretty much around. So if you've got a lot of ammonia, or what you can do with your pee, if you can go, because that smell that you get from when you do a pee is ammonia. So if nitrosomes is in the area, it wants that and NH4 to work with it to reduce it into nitrite. So yeah, I work with my pee, so my inputs are really good. I don't have antibiotics, etc., etc. So I know I can work with it. So I work with it, and then I know that this nitrosomus is being proliferated around those areas. So I already know I have that one. So by introducing nitrobacter, I've gone and fed my plants. It's pretty cool. That was a good slide, though. That should be saved. That's the last one, too. Where was that section? I'm just going to save that one. Copy. Put it on the main screen here, I suppose. Still sharing. But yeah, that was another section. That was really good reading that. Biopesticides. No, we're not in the pesticides. We're in the biofertilizer. Because you can use your pests as well, like your Bacillus thuringiensis or your, what's that other one? It starts with B. Anyway, but we're not talking about them today. It's about biofertilizer and how to make your microbes work for you to feed the plants. Plant microbiomes as contributors to agricultural terror, no. The pivotal role of guts, phytohormone producing ones. Molecular mechanisms underlying pathogen resistance. So that's, you can use your microbes also for that. Oh, here's a good one. Oh no, that's pesticides. Right. Talking about fertilizers today. Free. No, that's fungicides. That's bio. Talking about feeding the plant free. It's mineral elements. That'll do. Oh, gone way over an hour. Leaving it sit around, I've led to believe it stinks. Oh yeah, all caught up. All right. Is there anything anyone else wants to talk about, or is there any other biofertilizers we can discuss, or any ones you've heard of? How about the sides of the packets that you get in some of your those bios beneficials that you see from your shops? Have you got any ones on the side of that you want to go through? I still haven't thought of the Korean natural farming. That one they do. Just looking through a few more slides to see if there's anything here that's relevant for today. Fertigation, no. Probably go through. I'll see if there's another course, because I've done quite a few micro courses, but that soil one was one of the better ones, because that was written in 2022. So I'll share this and we'll just go through this and put your questions in if you've got any now, because this is pretty much wrapping up soon. I'm just going to finalise by seeing if I've got any other biofertilizer slides that I can add. Study. Diseases, that'd be biopesticides, biofertilizers. These ones will show it. No. Organic farming, that just touched into the different ones. Actually, there was. Then I'm going to go through them all though. So biopesticide section here. Vermicompost, yeah, pest and disease. Yeah, not fertilizer. Plant biochemistry, no. And that was the good one. I touched on it. Maybe this one at the top. Which one? Soil chemistry, this one. Oh no, that's more cation exchange, elements exchange, redox. Here we go. Inorganic soil organic matter. Okay, exchange processes, chemistries, pollutants, soil NPKs, retaining pollutants, modeling. No, that one didn't have any in it. Environmental soil chemistry. It was rad though. That was that's really important, this soil chemistry, to learning the exchange processes. So now I know how things can exchange, elements can be exchanged. Now I need to get the microbes to produce those elements so that they can become exchanged in the environments that are exchangeable. Because not all, like you get your different redox, your amount of oxygen that's in the substrates, and it diminishes so that the microbes can't perform its process because most of them rely on oxygen. So yeah, biofertilizers focus. To stay on topic. Cell biology, no. Cell culturing, no. That was how to do it all. Not on individual microbes. Experimental, no. There was different experiments you do to, like more so lab experiments. Biotechnology, so that's using microbes to remediate the environment, so you can use it to fix metals and pollutants. Doesn't look like it. I think that's it. I think that's it. Microbes. Microbes. beneficial bio-producing microbes. No. That's it. Fertilizers. Fertilizer, solid, liquid. No. Righto. Stop sharing. That's about it. That's it. Oh, look at that right at the end. Cheers. East Coast dankness. How you going, mate? You come right at the end. I just did a rad. We just went through heaps of different beneficial microbes and what they do and why you should have them in your substrates and if they're going to work together or not, if they're symbiotic or biofilm producers. But yes. Oh, mate, do you have any microbe questions? East Coast dankness. Today's topic was really biofertilizers, not biopesticides. So if you have any one specifically on that, that would be rad. Yeah. There was this page, the reason, because it was prompted the topic today. There wasn't really many questions, unfortunately, but there was because of this. So I've been going through microbes that produce phytohormones, and then they've also got secondary. They don't just produce hormones, they do other things too. So it's good to see what does what. And I went through and explained quite a few that what does what and why you should have them and how to make them yourselves, like these azos, or culture them yourself, etc., etc. I won't bore other people that have watched the show, but that's what I was going on about. So if you've got anything to do with that, throw it in quickly because the show's going to end. But it's just brilliant. We're learning to work with microbes and for them to work with you. Wow, what a win situation. And now they're just getting into the gut microbiome where they're saying our guts have that brain axis that goes straight to the brain, and they're figuring out that fat people have different microbiomes in their guts to skinny people and all this sort of stuff. Microbes, it's understanding them in their different forms and how they function. I think it's the win-win. And by this biofertilizer stuff, having the microbes engage when they're needed, it's sort of nearly like a remote control, or, okay, I want these plants to function well, press a button. They're always going to function well. It's, yeah, that's why I'm really into it. No, I'm trying to, how do you, I'll just write cool. Because then I'm just trying to put a monetarization, fair income. I'm trying to do, come on, add East Coast dankness. Oh, I'm going to show you what I'm doing. So you're going, what's he on about? All right, watch this. So to make East Coast dankness there, cheers. Look, I'm going to touch it. And look what comes up. So I want to go up here and press this button where you can go and put all this sort of stuff in there. And now look. Oh, I did it after all that complaining. Add as moderator. Manage standard. There you go. So it's just me and my weirdness, but I could share my weirdness. There you go. Why aren't I being made moderator? Now I've got to do the same thing again. Stuff that. All right. No real microbes coming through. Please bring your topics for next week's, because I've kind of talked about microbes for the last couple of weeks now, so I might have used that up for a bit. Have to change the topic. Yeah, so bring some different suggestions next week, please. Or actually, it was me. I made this up this week, so it's more so telling me. But I hope you can bring some suggestions next week, and we can make up a cool title and go through some more slides again. So I'd like to thank you all for listening, and I hope you have a fantastic week, and I'll be back doing the same thing again this time next week, in an hour or so, hour and a half early. Happy breeding, happy growing, and good health to you all. Bye-bye.